Okay, in this tutorial I'm going to talk about enzymes, a little bit about what they are, and essentially how they work. So an enzyme is what we call a biological catalyst. I'm going to talk a little bit more about what that means. So a biological catalyst speeds up a chemical reaction without actually being used up or being involved as a reactant in the reaction. So biological catalyst speeds up a reaction without being used up. That's basically the definition of a catalyst. Enzymes are proteins. They're a type of protein that either that essentially speed up chemical reactions as part of our metabolism. So an enzyme, well, how do they actually speed up a chemical reaction? Well, it's got to do with this idea called activation energy. So in a chemical reaction, essentially, often energy needs to be put in before energy is actually released. So I can show you that here. Essentially, in a reaction, even if it's exothermic, that means that it yields a net, or it ultimately yields more energy than needs to be put in. Often, energy needs to be put in to break the bonds between some of the reactants. When bonds are reformed to produce the products, often more energy is released than was put in. So even in an exothermic reaction, often some energy must be put in before the reactants, before bonds are broken, allowing them to reform bonds to form the products. And when those bonds form, often energy is released. So often some energy must first be put in. And that's what we're showing on this graph here, that for a chemical reaction, some initial energy often needs to be put in. And then once some bonds are generally broken, then new bonds can form and energy is released. And a lot of the time, the energy that is released can often actually exceed the energy that needs to be put in. However, some energy does still need to be put in to get the reaction to occur. And that's essentially what we call activation energy. Activation energy is the minimum energy required for a reaction to occur. And that's going to be different depending on the type of reaction. So a lot of reactions won't necessarily occur at room temperature. We might need to heat them slightly or provide some form of energy before the reaction will actually proceed, because we don't have enough energy to break those initial bonds. After those bonds have been breaking and new bonds form, energy may be released, and sometimes even more energy than we had to put in. That would be an exothermic reaction, and in that case then that might release the energy to set off more of the other reactants, or more of the reactant, and the reaction will then proceed all by itself. What an enzyme does is it provides an alternative reaction pathway. What that means is the reaction occurs slightly differently, and that alternative pathway may take less activation energy. So what do we mean by an alternative reaction pathway? Well, often in a reaction may actually involve a series of steps. For instance, again, if we come back to this reaction down here, the first step was the breaking of the bonds, and then the next step was the forming of the bonds between the product. And that first step required essentially energy to be put in to break those bonds. So in this reaction pathway, there are actually steps that we don't see necessarily when the reaction occurs. There may, for a very brief period of time, be, for instance, an intermediate. So the intermediate might be these molecules here, the reactants already broken apart before they have formed the new bonds to form the products. And these intermediates here may be very unstable, so they may only exist for a fraction of a second. But when our enzyme provides an alternative reaction pathway, these intermediates may be different. They may somehow be associated with the enzyme. So, back to my activation energy. Essentially what an enzyme does is lower the activation energy. It lowers the amount of energy that is required to get a reaction to occur. So it lowers the amount of energy that we require for a reaction to occur. And let's look a little bit about how can it actually do that? How can it lower the energy that's actually required? Well, we've got here a reaction without an enzyme. So here we've got two reactants. The blue substance here is one of our reactants, and the red substance is another reactant. Alright, now what you can see here, some of the product has formed. The product being the blue and the red stuck together, a bond has formed. So in this reaction, the red and the blue are essentially reacting together to form this product here, the red and blue square stuck together. Now what you can see is that normally, without an enzyme, the reactants will essentially be moving, and this is essentially what we call collision theory. And when two of the reactants collide with enough energy and with the correct orientation, a collision will be successful and product will be formed. So here that first collision didn't quite have enough energy or didn't quite have the correct orientation. So we can speed up reactions by, for instance, heating them, which would make the particles move faster. If the particles move faster, we'll get more collisions and more successful collisions. But another way we can speed up the reaction is by providing a better orientation for the reactants. And this is essentially what an enzyme does. So with an enzyme, the enzyme here, which is a type of protein, so this enzyme is made out of a long chain of amino acids all folded up into a really precise sort of 3D structure. But this enzyme will essentially bind temporarily with the reactants and provide the orientation needed for a successful reaction to occur. So this enzyme here essentially provides the orientation to increase the likelihood of a successful reaction. And this is essentially what this means is that we can substitute energy rather than just heating up the mixture so that we get a larger number of collisions or collisions with greater energy that are likely to be more successful. We can substitute energy with orientation. So if we go back to my other diagram here, what happens is we need less energy because the substrates, the reactants, are more likely to collide with the correct orientation. So back to, we said that the key thing an enzyme does is it lowers the activation energy, the energy needed for a reaction to occur. And it does that by providing an alternative pathway, reaction pathway. And for instance, that often involves the enzyme temporarily binding to the reactants, and essentially providing them with a more correct or a better orientation, so increasing the chance of a reaction occurring with less energy present. And that's why this energy can proceed with less energy. Now, you could imagine here there's energy in the ambient environment. The room temperature provides energy, and you could imagine that with the yellow line, without the enzyme, room temperature may not be high enough for this reaction to occur. There may not be enough energy to kick the reaction off. Whereas with an enzyme, this green line here, because suddenly we don't need as much energy, it's giving it that orientation instead. At room temperature, if room temperature say was here, suddenly there may be enough energy for the reaction to occur. So even with an enzyme, usually some energy is still required for the reaction to occur. But what it means is the ambient temperature, room temperature, or whatever temperature the enzyme operates under, that might be enough already for the reaction to then proceed. And what this means in practice, in real life, is that a lot of reactions will occur with an enzyme, and without an enzyme, basically they occur so slowly that by all practical means, essentially, they don't really occur at all. So that's what an enzyme does. Speeds up chemical reactions by providing an alternative pathway and reducing the activation energy, the energy needed to get the reaction to start or the reaction to occur. So guys, I hope you found that one helpful, and I'll try and get on with the next one.